Hi, I'm Alex Driehaus. I'm a physics and math major from Western Kentucky University. And this summer I was working at A&M with Dr. Siebert on the effects of direct low temperature plasma treatments on the germination phase of feminized industrial hemp varieties. So I'm going to give a little bit an introduction as to my experience with the ALPIP program. Then I'll be giving a brief introduction to plasma, and then plasma in agriculture and just some research that's happening at A&M. Then I'll talk a little bit about hemp. Then I'll talk about plasma and hemp, kind of joining the two, and then a little bit about the research station. So from this program, despite the fact that it seemed kind of isolated when I was looking at it, it actually turned out to be an experience that was very interdisciplinary. It was very interactive. So we did interact with the REU group. Even though I worked at A&M, I was housed at UAH. We also used the plasma lab at UAH and UAB. So it was a really good opportunity to kind of see everything that was happening across the board in plasma in Alabama. So we're going to talk a bit about plasma and just general plasma applications, which include your natural plasmas like lightning and the sun and space weather. Then you have microwave plasmas, and it's used in sanitization in clean rooms and for manufacturing to clean equipment. We focused on low temperature plasma, which is generated with a high voltage source, which has to be in the kilovolt range. LTP is only surface deep, so a lot of the plasma you can actually touch because it's only going to really reach through the top layer of your skin. You can treat water with plasma, and that'll add nitrates. And there are a lot of forms of plasma instruments in the low temperature plasma realm, which include sheets, jets, and chambers. When we use plasma at UAH, we used helium to generate the plasma, and we used a sheet and a jet as shown here. The sheet is the top right. We're treating hemp seeds, and then the lower left, I guess that's a jet, and that is treating a semen extender for cattle. Their plasma is generated at 8.17 kilovolts and about 10 milliamps. When we went to UAB, we used their chamber. We used nitrogen to generate the plasma. It's a pressurized chamber, so it operated at about 0.998 torr. And one main difference, other than the pressurized aspect, is that they actually bought the unit from Herrick Plasma, whereas UAH designed and built their plasma instruments. So plasma agriculture at A&M involves some work with turmeric. There's plasma treated water within that. I'll talk a little bit about the turmeric later. There's also a look at plant health with plants that have known microbes and bacterial and fungal cultures. It's mostly sanitization. And then there's some food safety issues that they're also looking at, which are mostly sanitization of fresh meats and vegetables. So a brief talk about hemp. Hemp is cannabis, and it has two main components that a lot of people are focused on when they talk about hemp. This includes THC, which is psychoactive, and the other is CBD, which is non-psychoactive. And CBD actually is a wide range of compounds that just fall into the cannabinoid category. They're all non-psychoactive, and that's where you get the pain-killing benefits. Most hemp varieties are dioecious, which means that you have a female plant and a male plant. But there are some monoecious varieties where you have male and female attributes in the same plant, although monoecious varieties are all selectively bred by humans, and they revert back to dioecious tendencies in later generations if they're allowed to interbreed. So THC is a really big deal in the world of hemp for a number of reasons. One of them being that that is what defines industrial hemp from marijuana. So industrial hemp, by definition, has less than the legal limit of THC. In Alabama right now, that is 0.3%. And this is strictly held. So we actually got all of our feminized hemp varieties from Oregon State University. And there was one that we couldn't plant because its THC level was 0.3007%, so less than 1/1000th of a percent too high. And once it crosses that 0.3% mark, it's considered marijuana. So then all of the crops of that type have to be disposed of according to government regulations. So one way to obviously differentiate hemp is industrial or marijuana. But you can also do it by feminized or fiber varieties. So feminized hemp is mostly female. One thing that's studied widely in hemp is the sexual nature of the plant. So even if a plant is chromosomally female, that doesn't mean that it will show only female attributes. You have some that are chromosomally female that show only male attributes, and the other way around. So they're mainly female. One male plant can ruin an entire crop of feminized hemp because once they're once they're pollinated, all of the buds are turned into seeds, and the buds are what contain the CBD, which is what you grow feminized hemp for. So fiber hemp, because you're growing it for the plant, not necessarily the THC or CBD, you can grow females or males. You don't really care which one. So it's about a 50-50 mix. And usually they're cultivated to produce materials like paper, hempcrete, and fabric, and other stuff that falls into that material category. So historically, hemp was one of the first domesticated crops by humans. So it's been grown for millennia. It was grown in the 1600s in Georgetown. It was the paper that the Declaration of Independence was written on, and the first draft of the Constitution. And most of the founding fathers grew hemp. So it has a really rich history in America. In the picture on the right, that is a field of feminized hemp in between pecan trees. It just has a very lengthy history in the US, which was then heightened during the war on drugs. So it reached its peak at about in the 70s, which is when they designated marijuana and hemp as a Schedule I drug. And so any research on the plant was lost in the US. Any research that you find between the 70s and really today is going to be international research. The USDA keeps seed reserves for any crops that they grow. And during that time, they destroyed those seed reserves because it was considered a dangerous substance. So now that it's becoming more legalized, they're trying to replenish the research that they could have done during that time period. One thing they're doing is rebuilding the seed reserves. That's a project that Cornell is working on, and A&M is now becoming a partner with them to help with that effort. And so growing industrial hemp is getting to a point where it's easier, but depending on the politics, it can be difficult to do still. This is just a graphic that represents some of the modern uses for hemp. And as you can see, they use every part of the plant, just about. So you have, you can use it for your textiles, your building materials, or you can use it for medicine. You can use it for animals. One thing that they're doing here is testing it as a feed for chickens during the Skip a Day program. So it has a lot of uses, and you're using them holistically. So now for our project, we're looking at plasma and hemp. So we were looking at whether or not plasma increases the germination speed in hemp seeds, the difference between feminized and fiberized seeds when a plasma is applied to them, how long is the optimum exposure time for plasma treatments, and is there a difference between atmospheric and vacuum plasma? So for the first trial, we went to UAH. We treated 50 seeds from each of 8 cultivars. The feminized cultivars we used were Rogue, Alpha Explorer, Queen Dream, and Photo CBD. These were all sent to Dr. Siebert and A&M from Oregon State University. We had 4 different treatments. So we had dry untreated, dry treated, soaked untreated, and soaked treated. And the soaked varieties we soaked for 24 hours before the treatment in deionized water. And we treated all the treated seeds for 60 seconds. We germinated the seeds from trial 1 in Petri dishes. We found the fastest germination time in the soaked treated seeds and the slowest in the dry untreated. But across the board, the soaked seeds did better than the dry seeds. This is a graphic showing the distribution of those treatments and how they did during the last measurement. So on the right, you can see the mean distribution and the error bars that are associated with it. And then on the left is a Duncan grouping based on that distribution. So in the Duncan grouping, any varieties and treatments that fall within the same bar are statistically insignificant, any differences between those. So for this one, feminized for the soaked treated, soaked untreated, and dry treated are significantly different than the dry untreated from that same variety. So across the board, the soaked seeds did better. And between the dry treated and dry untreated, the dry treated tended to do better. After we germinated the seeds, we didn't just throw them away, we planted them in the shade house, and then we transplanted them into raised bed gardens. So for trial 2, we looked back at what we did for trial 1, and we kind of edited the process. So for the first trial, we took average masses before and after germination, but just to see the distribution, we decided to take the mass of each individual seed, and we germinated them in the vials that you can see in the upper right. So that also let us keep track of the seeds through the measurement period, because in the Petri dishes, they would grow and kind of curl around each other, and it got really easy to lose track of which seed was which. For this one, we used 100 seeds from each of 6 cultivars. The feminized cultivars were Rogue, Alpha Explorer, and Queen Dream. We only used 3 because we found that there were similar trends throughout all of the cultivars that we used for the first trial. So whatever we see in those 3, we would have seen it in the other one as well. We treated half of the seeds at UAB and half at UAH. The UAH seeds were treated by the jet, and the seeds at UAB were treated in a chamber, and we were testing optimum exposure time. For the first trial, we treated everything at 60 seconds. For this one, we did 30-second increments up to 120 seconds. At UAH, this picture is how we treated them. We treated them in the little vials with the plasma jet. This is again helium. We treated 1 seed at a time just to maintain the seed number throughout the process. We treated the soaked seeds about a day before all the other seeds. At UAB, we modified these trays for treatment so that we could treat all of the seeds from each time sequence at the same time. So we cut them right here. I don't know if you can see my mouse. So we could fit them within the chamber, and we didn't have to do each seed individually just to speed up the process. For this, we again took 3 different measurements. This time, again, we had the individualized data. So just as an example, this was the Rogue variety. And we found that by the graphic on the right, the post-germination weight was highest in the dry untreated and dry treated 90, and those 2 were significantly different from the soaked treated 90 seconds. And by the left, the soaked treated 120 was significantly different than all of the other varieties. So that one, except for the 60 seconds. So that one is the better exposure. And just to see how it fits into all of the treatments, this was, again, a combined feminized fiber chart, and it shows that, as we saw before, the soaked seeds tended to do better than the dry seeds, and the treated seeds that were dry tended to do better than the dry seeds that were not treated at all. During this time, we also treated some barley, and we sent it to Hartwick College in New York for an alpha amylase analysis. This is a compound that is vital in germination. So we tested to see if the plasma increased the alpha amylase in the barley. We treated them at UAB for 60 seconds. Again, we had the soaked untreated, soaked treated, dry untreated, dry treated. And we actually found that the soaked untreated germinated earlier than the soaked treated, which was not in line with our results from the first trial. So we then took some more barley and treated it at UAH for 60 seconds. And we found that reinforced the trial 1 effects, and the soaked treated germinated the fastest. Just as a visualization of what the plasma actually does to the seeds, these were some hemp seeds that we treated for an alpha amylase treatment, but we couldn't find a place to send them for that analysis. So on the left, they're untreated, and on the right, they're treated. So you can see that the plasma made them darker, and the veining got more distinct. They almost look like turtles, is how I kind of want to explain it. But that's just a visualization of the scarification from the plasma. This was the actual data from Hartwick College for the alpha amylase in the barley. And we found that kind of like we expected, the soaked did a little bit better than the dry, but once we put them in the chamber, because of the vacuum, it sucked a lot of the moisture out of the seeds that we had soaked. So that kind of made the results different than what we thought they would be based on the atmospheric plasma. So our conclusions are that soaking is beneficial across the board in the germination of hemp seeds and barley seeds. Atmospheric plasma seems more beneficial than vacuum plasma, at least for these biological cultures. The treatment of dry seeds seems to increase the germination speed. And though the treatment of soaked seeds seems beneficial, we'll still need more testing and study in order to actually decide definitively whether or not it's better. So one thing that's different from all the other projects mostly is that this was a completely different environment. A lot of the other projects were at a lab with an engineer or a physicist, but we were working at an agricultural research station. So, like I said before, this wasn't as isolated as I thought it would be, but even within the research station, we weren't working on our isolated project the whole time. So I just wanted to give an overview of some of the other projects that were happening that we helped out in while we were here over the summer. This is Dr. Siebert working in the field and planting some beans. So one project they have going is the Circle of Sustainability. So they're growing tilapia in these tanks in the shade house. On the left, there are raised beds, and they have plants for consumption by the tilapia. And in the right, on the back, you can barely see it, but that is food for human consumption. They're using the waste from the fish, they're filtering it, and the solid waste goes to fertilize the food for fish consumption, and the liquid waste goes through a hydroponic system to give nutrition to the food for human consumption. So it's one ecosystem where nothing goes in, and you know exactly what the food you're eating, where it came from, what went into it. This is barley for beer. This is a project that they have with Hudson Alpha, Auburn, and North Dakota State. Auburn bought a malter, and they have hops. So A&M is testing the malt efficacy of 25 different cultivars of barley to see if that would be a good crop for local farmers to grow for local breweries, to see if you could have truly local Alabama beer. This is turmeric. Like I said, I would talk about this later. Mr. Lam is a PhD student at A&M. He is from Vietnam, so he brought with him 52 different varieties of turmeric. Previously, turmeric hadn't been grown in the US further north than Florida, because in a tropical climate, it takes 9 months to grow in a field because it has a really long dormancy period. So with the use of the plasma, they cut down that dormancy period, and it'll grow in a time frame that's conducive to Alabama climate. There are also cows. Dr. Bob, who is in the bottom left, he works with the cattle. One thing they're doing is they're testing the stress in cows due to separation of the mothers from the children, or I guess the cows. And they're also testing the stress from heat, because it's hot in Alabama in the summer. And so they're testing the treatment of melatonin in the cattle. They also had us treat some semen extender with plasma to see if that will extend the life of the semen extender. One big project here is miscanthus. They're working with Hudson Alpha. It's a biofuel project, or at least it started as a biofuel project. Miscanthus was used because of the large biomass, They cut the miscanthus every year. This is the second year it's grown. And in March, you could drive a little tractor in the field of miscanthus, and now it's taller than everyone who was there. Right now, they're looking at moving from biofuel to studying fibers. So I would like to thank Dr. Siebert, Dr. Kwong, Ms. Michaela, my partner, Mr. Lamb, Dr. Bob, Dr. Mintretti, and the faculty and staff of the Winfred Thomas Agricultural Research Station, Dr. Shu, and the faculty and staff of the Propulsion Research Center at UAH, and Dr. Vorha and the faculty and staff from the Center for Nanoscale Materials and Biointegration for their services and guidance during this program, along with the NSF EPSCoR CPU2AL ALPIP program for allowing the internship to take place. And these are my references.
Hi, my name is Plasmy, and thank you for watching this video. Make sure to follow us on our social media at CPU2AL on Facebook, LinkedIn, and Instagram. Bye!